everybody. Thanks for being here for Science at the Edge. So I am your moderator. My name's Clarissa. I'm from Kickstarter, and I handle science outreach for Kickstarter. And I have the great pleasure of moderating a discussion with some of our really exciting panelists. And I think we're going to kick this off with a few short talks, and then we will go ahead and jump into a discussion. And I have a few questions of my own to ask our panelists. And then we're going to open it up for Q&A from you guys. So yeah, and we're on a time schedule, so I'm going to just kick it off and let our first panelist come on up and give a talk. Yeah, please, welcome her up.
That's me. I'm Mary Hagedorn. I'm at the Smithsonian Conservation Biology Institute. And it's a real pleasure to be here today, so thank you to all the organizers. I first started thinking about the Earth in grammar school because we would watch early space explorations on television in grammar school. And I actually never really figured out how this worked because it was an inner city Catholic grammar school that barely had money for gym and lunch, as far as I could tell. I don't know where they got the money for televisions in every classroom, but it really got me fascinated with space. And this image that Bill Enders took, Apollo 8 astronaut, of Earthrise from the moon, really changed my view of the world. I never understood really how incredibly beautiful the world was. And every time I see the incredible images from space of the Earth, it just inspires me because it is so incredibly beautiful. Today, our lust and our greed for fossil fuels is changing our world in ways we can't even anticipate for the future. And one of the things that sort of keeps me up at night is wondering how the people and the animals that are going to come after me will live on this Earth, and whether the beauty that we see on the Earth now will still exist. So one of the, obviously taking on a whole planet in terms of conservation is a bit beyond, is more like Star Trek and Star Wars. And I tried to limit my interest to just saving coral reefs. And that's what I'm going to talk about today. So the question I wanted to pose to you today is, can someone who has some rather unorthodox ideas about conservation really help save coral reefs? And we'll see what you think at the end. So I want to tell you a little bit about why coral reefs are important and what they do for us. 50% of our oxygen, we live on an ocean planet, 50% of our oxygen comes from the ocean. Our coral reefs are one of the most important ecosystems in our ocean. A quarter of all animals that live in our ocean at some point live on a coral reef, so they're major nurseries for our oceans. They're also important for our cities and our homes. I live very close to a coral reef, and we get tsunamis in Hawaii, and I'm very grateful for that barrier reef that protects my house. They're also important for antibiotics and new pharmaceuticals that places like NIH are looking at, because they've been around for millions of years and have chemical defenses much like trees do in our forests. At least 500 million people depend on them for their livelihood, whether it's fishing or tourism, et cetera. So they're really important in terms of our economy, especially since about $350 billion annually comes from coral reefs. But the global problem, and there are local problems, but I'm going to speak mostly about the global problems. The global problems is that every single coral reef on Earth is under siege. No matter where it is, whether it's near people or not near people, it is being affected by global climate change. And part of it is our greed for fossil fuels, producing carbon emissions that both warm our oceans and cause acidification of our oceans that leads to coral stress, coral bleaching, coral disease, and potentially death. And so this triage, or this horrible sort of cascade of events can happen to coral, especially when they're stressed by local things like pollution and sedimentation, et cetera, et cetera. One of the things that we do know, that if coral reefs start to fail, hunger will increase, because so much of our population, 1/7 of our population, depends on fish for food. And without coral reefs, fish decline. Here's the really bad news. 2/3 of the Great Barrier Reef is dead or in danger of dying today. A year ago, I never would have said that, but it's astonishing to think about that almost 1,000 kilometers of our Great Barrier Reef is now under siege. And it happened quickly. It's been stressed, but it happened very quickly. So the question is, what can we do about this? How can we move forward and be optimistic about this? So a baby is probably an odd transition to go from smokestacks and dying coral and climate change, but it's an image of hope. It's an image of sort of the future, and it's also what we do at the heart of our science. We use human fertility techniques to save coral reefs, and we know that about 5 million people on Earth have been created with in vitro fertilization. And we're using exactly those same techniques for our work. Now, when you look at cryopreservation, or the freezing of human gametes, it started out with sperm, because sperm is relatively small and easy to cryopreserve. And then came, and that was in the 1960s, and then in the 80s we had human embryos, and then not until about 2000 did we get really good cryopreservation of human eggs. A lot of that has to do with them being bigger, eggs and embryos bigger, and eggs are much more complicated than embryos. They have a lot of eggs in them, a lot of yolk in them, and they're just harder to freeze. So when we cryopreserve something, it is with the idea that we can safely freeze it and thaw it. And we introduce a cryoprotectant, which is an antifreeze chemical, and we try to avoid ice crystals, which you see in this picture here. And it is the avoidance of ice crystals that is critical in safe cryopreservation. And we do this experiment all the time at home. If you wrap your meat or your vegetables or whatever it may be insufficiently, the freezer will go through thaw and freezing cycles that will produce ice crystals that give you freezer burn. And it becomes unpalatable at that point. So it's very similar to the damaging actions that happen during bad cryopreservation. But what I want you to think about in terms of cryopreservation is that and what we are doing it with is that we are doing something similar to a seed bank. We're creating a repository using human fertility techniques to cryopreserve coral and for the future. And it's very similar to a seed bank. Seeds can last for tens to hundreds to even thousands of years. And frozen tissue that's well maintained can do the same. We use seeds to enhance our ecosystems, and we can use them to bring back extinct species. This is exactly what we can do with frozen repositories as well. So coral, I just put the pandas up here, mostly to show that pandas reproduce in a very restricted amount of time, as do coral. Coral only reproduce for about 2 nights a year, and it's 40 minutes each night. So we have a very restricted time to work on them. So what can we do, and what are the impacts of our cryopreservation? We can freeze coral sperm, and we've developed very inexpensive techniques to do that with. We can take that sperm, we can thaw it out, and we can create new coral. You see some on the left is from fresh sperm, on the right from cryopreserved. They're virtually identical. And now we've banked over 16 species from the Caribbean, Hawaii, and the Great Barrier Reef. And we know that if we can take 35 individuals, we get 90% of the genetic diversity in that area. In my laboratory this summer, we just successfully cryopreserved fish embryos, which are very large, very similar to human embryos, but a much larger scale, using gold nanoparticles and lasers. And the lasers were there to stop ice crystals from forming. We'll be using that same technology to try and cryopreserve eggs and embryos, and I think we'll be very successful this coming summer. these are the kind of techniques that people want to use if they're going to be doing hybridizations, selective breeding, and doing extensive genetic work. They want to be able to mix and match sperm and eggs. The thing that's happening right now is that bleaching is impacting reproduction, and it is decimating the future of our coral and our coral being able to adjust to new conditions. So we're taking a slightly different tact. We're trying not to focus on reproduction over the next 3 years, because in the last year, all of our efforts in Maria and over reproduction has ended up with 0. We got no reproduction over this last year. So we're now freezing the algae inside the cell, inside the coral, which are called symbiodinium, and we've successfully cryopreserved those and hope to make a global bank for those, because these help control the resiliency of the coral. We're working with Ken Niedermeyer and creating offshore nurseries, and he started these in Florida. We're putting them in Hawaii and Moorea. This is what they look like today. They're no longer just these cement blocks. But these nurseries are causing corals to grow faster, and they're reproducing enormously in these nurseries. So they're really an extraordinary tool, and we're making them for Moorea. But then finally, we're working with Mote Marine Lab and Dave Vaughn there, and they're making these tiny little microfragments. And the microfragments, we can get them down to 1 polyp. We've already successfully cryopreserved fragments of coral. We need to enhance it and make it so that it's conservation ready. But we hope to put these microfragments, grow them out quickly on the coral trees. So we're going to be putting both reproductive material and these microfragments. If we can get the microfragments to really work in terms of a conservation practice, we will no longer be hemmed in by 2 days a year. We can go through the reefs quickly, identify the number of individuals that we have, and know the genetic material that we have, and we can train people very quickly to do this. So we think that this is going to be a really amazing tool for successfully trying to maintain the biodiversity of reefs. So I told you coral reefs are in trouble. We have this amazing bank that we're forming that we hope people will start using today, but will be there for tomorrow if in 500 years the oceans have returned to pre-industrial conditions. And we're hoping, though, that today these important tools that are ex situ conservation, or however you want to think of them, can be incorporated into the conservation strategies for larger conservation actions like half the planet conservation, whatever it is. So because we think we can maintain the biodiversity of coral reefs. So I'll leave you with this quote from E.O. Wilson, who I think gets the heart of everything that we're doing, and acknowledge my funders. Thank you.
Good afternoon. Thank you all for coming. So I'm going to give a talk that builds very much on the talk that Mary just gave. So thank you for such a great talk, Mary. I'm going to be talking about some other coral reef conservation technologies. And I'm a coral reef ecologist at the Scripps Institution of Oceanography. And I got into coral reef science because when I was an undergraduate, I was able to dive on the Great Barrier Reef for the first time. And just when I dove into the water and saw the beauty and diversity and complexity of coral reefs, I was hooked. And I've been studying coral reefs now for 20 years. And unfortunately, as Mary explained, a lot of the reefs that I've been working on and reefs globally are in a state of decline. So I wanted to dedicate myself to finding ways that we could protect reefs and hopefully take some of these damaged reefs and find ways to make them look more like the beautiful and diverse reefs that we still have in several of the remote parts of the world. So today I'm going to focus on conservation technologies. I'm going to introduce you to a few of the technologies that we've been working on, and I want to emphasize how important collaboration has been on these projects. So with all of the projects I'm going to present today, they've involved biologists, chemists, engineers, and computer scientists to really try to develop new approaches for trying to better understand and protect coral reefs. So the first one I want to talk to you about is what we call the Computer Vision Coral Ecology Project. And when we survey the reefs of the world, the normal way that we do that is we take underwater photographs. We do it along transects, so we swim along these lines, taking photographs every couple of meters. And then the hard work begins when you have to take these photographs back to the lab and try to get data from the photographs. So in a mission, you could collect hundreds to thousands of photographs. And unfortunately, less than 1% of all the photographs taken of coral reefs for coral reef science and conservation are ever analyzed. So we teamed up with some computer scientists at the University of California, San Diego, and using computer vision and image recognition technologies, a lot like the technologies that are in Facebook. So when you click on photos in Facebook and you tag your friends, after a while, the computer starts to recognize your friends' faces. Well, what we're doing is we're tagging photos of corals and training the computer that this is coral A, this is coral B, this is algae. And after a little bit of training, the computers can speed up the process. So we call it a semi-automated process. And with this process, we can analyze the photos and get data from them 10,000 times faster and at over 90% accuracy compared to coral ecologists doing it manually. So we're hoping that by using these computer vision technologies that we'll be able to expand the scale of coral reef surveys and coral reef health data, and also really make the data, get the data much quicker so that we can make better conservation and management decisions. And another thing that we've been doing to try to expand on what we're doing with coral reef surveys is we're working with a robotics group in Australia that uses underwater robots, or AUVs, that image the reefs with calibrated stereo cameras. And rather than just getting these snapshots of what the reef looks like, we can image several football fields of reef at a time down to great depths where human divers can't go. And these 3D photo mosaics even give us information about how three-dimensionally complex the reefs are, which tends to be a really important parameter for coral reef health. And by combining this technology with our computer automation technology, we're working on trying to automate every single pixel in a photo mosaic like this, so that if we do repeat photo mosaic surveys, we'll actually be able to get new important ecological information, such as the rates of growth and mortality of all the different organisms on the sea floor. And we feel like this will really revolutionize how we do coral reef ecology and the sorts of information that we can use for conservation and management. So the next project I'm briefly going to talk about is we wanted to see how all of these climate change impacts that Mary mentioned, how they're affecting coral reefs. So I worked with a team of engineers in Australia and at Mbari, and we developed what we call an underwater time machine. And this system, which we call our free ocean carbon enrichment system, it uses a sensor array to actually produce future levels of CO2 actually on the reef itself, with all the natural ecosystem and all the associated organisms. And we're using this time machine to be able to understand which parts of the reef ecosystem are most susceptible, who are going to be the winners and losers of climate change. And we're trying to develop now a portable version so we can even figure out which parts of the reef are most vulnerable, which are most resilient, and how we might be able to change our management strategies to take some of these considerations into management plans for a future in a high CO2 world. And then the last project I want to talk to you about is what we call CSI Coral Reefs. And this is a collaboration with Martine Tresgueres at Scripps. And Martine is a cellular biologist, and using medical methods that are commonly shown on CSI, where they're trying to figure out who committed a crime, we're using them to actually figure out which cells and which cellular processes are involved in coral reef stress. So when we can figure out where the stress reactions are occurring and get the mechanisms of how corals deal with stress, we're hoping that we're going to figure out new methods for trying to minimize the stress that the corals are experiencing. And I want to close by saying we've seen a lot of great talks in the last couple days about how science alone is not enough. And I feel like one of the only ways we're going to save coral reefs, considering the scale of the problem, is through education. So we've been working on a project with PBS and Jim Henson's Splash and Bubbles, which is a kids animated program for K through second graders. And we're developing educational materials to get young children excited about the oceans and about protecting their oceans. And then just last month, I was on a panel with Jean Cousteau about a new biopic of Jacques Cousteau, a new French biopic. And this, seeing the film and being part of this panel made me realize what an important character Jacques Cousteau has been for the oceans. He's inspired millions to care about the oceans, and he inspired me to become a marine biologist. And I feel like we need to collaborate with creative types, with filmmakers, with photographers, with musicians to build a narrative. We heard Randy Olson's talk about the importance of a powerful narrative. Jacques Cousteau was king of creating amazing narratives around the oceans, and we need to make these narratives around coral reefs. And I'm going to be working on a documentary about the Cuban coral reefs, and we're actually doing a docudrama where there'll be actors that are along on a coral reef expedition where we document the reefs. And we're also working on a series of documentaries and potentially television shows where we show the reefs using virtual reality and underwater microscopes to give a new perspective on coral reefs and try to highlight some of the success stories of how we protect coral reefs. And thank you very much for your time, and I hope you'll join me in the mission to try to protect coral reefs for the future. Thank you.
Thanks very much for having me. My name is Brian Greatwork. I am the amphibian conservation biologist at the Smithsonian Conservation Biology Institute. 1989 was the last year that the golden toad of Costa Rica was seen in the wild. It is also the last year when scientists coined the term enigmatic amphibian declines to describe mysterious disappearances of amphibians from all over the world, including in national parks. Amphibian biologists investigated many different causes of the declines, but it wasn't until a decade later that a smoking gun was found. Researchers in Costa Rica, Australia, and the National Zoo independently discovered a new skin pathogen associated with the deaths of frogs and called that disease Radiomycosis. The amphibian chytrid fungus has now been linked to the declines and extinctions of about 100 species of amphibians. In Panama alone, 9 species have disappeared from chytrid-related declines, including this Cherokee Harlequin frog, which was last seen alive in 1996 on the Costa Rican border with Panama. We have now recommended to the IUCN that they officially list it as extinct. Herpetologists were concerned that the iconic Panamanian golden frog might also disappear, and they created ex situ assurance colonies of this incredible animal just in case their predictions came true in the year 2000. And then just 6 years later, in 2006, the amphibian chytrid fungus arrived in El Valle de Anton, which is the home of the Panamanian golden frog. And there were dramatic amphibian declines, resulting in the loss of 90% of the individual frogs and about half of the species in a community in just 5 months. The last individual Panamanian golden frog was seen in the wild in 2009. Fortunately, the captive breeding colonies were a success, and now 2,000 of these frogs exist in captivity, including at the National Zoo. This encouraged zoos and aquariums all around the world to try and expand this concept and to build a global amphibian ark. In 2008, we formed a consortium of zoos and aquariums, including the Houston Zoo, the Cheyenne Mountain Zoo, Zoo New England, Defenders of Wildlife, and the Smithsonian, and we teamed together to create the Panama Amphibian Rescue and Conservation Project. Our mission is to build capacity in Panama to house and care for the most endangered species and to search for a cure for the disease that's been wiping them out. So Maersk Line donated 7 refrigerated shipping containers to us, the same kind that ferry ice cream and frozen strawberries all around the world, including up and down the Panama Canal right next door to our facility. And we modified these shipping containers to breed critically endangered frogs and keep the chytrid fungus out in a biosecure environment. We considered all 214 species of amphibians that live in Panama and determined that our chances of actually avoiding an extinction were highest for just 12 priority species that we now care for in these 7 shipping containers. So the zoo research and conservation community over the last 20 years has learned a lot about managing small captive populations. And this has really allowed us to build on the successes of previous efforts like black-footed ferrets and golden lion tamarins to use these incredible tools that have been developed, like stud books and assisted reproduction techniques like Mary was telling us about, and frozen sperm biorepositories to manage our small populations so that we can prevent unintended bottlenecks, genetic bottlenecks from captivity. And if all goes well, we get baby frogs. And to riff off Elder Leopold, this ensures that we have the necessary cogs and wheels so that we can begin tinkering to get these animals back in the wild where they belong. This week, we announced our most recent cause to celebrate. This is the first ever baby captive-produced vanishing robber frog in captivity. And it's called the vanishing robber frog because it was described pretty recently from a park where it had already disappeared. And furthermore, the 8 most closely related species to this vanishing robber frog are already thought to be extinct. So this is a cause for celebration. And we've spent a lot of time really trying to get a handle on ways to fight the disease. And in the last few years, microbiologists have significantly advanced our capacity to observe microbes using new molecular tools without having to culture the bacteria on frog skin first. And when we looked at the microbiomes of Panamanian golden frogs, we found that a few special captive-bred Panamanian golden frogs were able to become infected with the chytrid fungus and totally clear the chytrid infection. And they all had a very unique skin microbiome. And the key distinguishing bacteria on the survivors were not found on the frogs that died. So over the last several years, we've spent a lot of time isolating antifungal bacteria from frogs and then trying to add them to experimental groups of frogs and then expose them to the chytrid fungus in an attempt to develop a probiotic cure. And it hasn't really worked very well. So we said, we're going to be cleverer here, and we're going to take the most common skin bacterium found on all Panamanian golden frogs in captivity. We managed to isolate that and culture it, and then we genetically modified it to produce violacine, which is a very strong antifungal compound. And we successfully did all of this and put it back on the golden frog skin, but unfortunately, it seems like frog skin microbiomes don't like to follow our instructions, and they had no protective effect. In addition to the microbial defenses, host immune responses are also associated with different disease outcomes. New transcriptomic tools allow us to compare the genes that are most active in frogs that resist chytrid infections and to compare them with the transcriptomes of frogs that die. We even looked at the transcriptomes of those same surviving golden frogs to see if they had any special genes, but it turns out that because we took our transcriptome samples after they had cleared infection, they'd already returned to their baseline genetic levels. So what we're now doing, Alyssa's in the room here, she's the one who's doing that, she's the culprit, is trying to develop a tool that will allow us to just swab the same individual or take a skin biopsy from the same individual during the course of an infection. So we can actually look and see which genes are being turned on and off and see how that relates to a specific gene outcome, a specific disease outcome. Now, studying diseases in wild frog populations is very challenging. Most of the frogs in any community are just too rare to get sufficient sample sizes. Studying multi-species epidemics is always very much a case of now you see them, and now you don't see anything, and you're just left with a frustrated researcher in the field with no frogs to study. So our success with captive breeding has now allowed us to produce enough surplus frogs that we can begin our first experimental releases of large numbers of Lamosa harlequin frogs that we'll be doing this spring. And we will track these using ultra-small radio transmitters that weigh just 0.3 grams. And for the first time, we'll be able to observe the fate of a discrete cohort of a known number of frogs of a single species and try and get more detailed insight into disease dynamics. But one of the most rewarding aspects of this work has been the tremendous outpouring of public support from zoos, aquaria, donors, foundation, and the people of Panama. Every year we help to coordinate a Golden Frog Festival, attended by about 3,000 people, and there's an annual parade where all the kids dress up like golden frogs and march down the streets of El Valle de Anton and take pride in their national symbol. Our project is ambitious, and it's one of the largest amphibian conservation efforts of its kind. Because we were willing to tackle the problem at a national scale, 12 incredible amphibian species now have a very different future. While our efforts feel Herculean, they're just a drop in the bucket when placed in the context of the global need. That is why the Amphibian Survival Alliance has been created, and that's going to help us to build the capacity and the partnerships we need to scale up our actions to have an effect at a global scale. It's easy to be optimistic about conservation stories that have already achieved their goals. Those congratulations are hard-earned. But a vision for the future in the face of tremendous uncertainty is most needed at earliest stages in the project cycle. And I'm optimistic about the future of amphibians because I can sense a new momentum, building a virtuous circle. Each new win and breakthrough and discovery inspires new questions, it motivates new actions, better husbandry, new funding, and it attracts new talented colleagues to help try and solve the problems. And on that note, I'd like to thank all of our employees, collaborators, and partners for helping us to realize the vision to preserve Panama's rich biodiversity heritage.
¡Rescatando los anfibios!
I'm not so good with Macs, so I can maybe get a quick hand. Is it just the next slide, or can you scroll down?
Sorry, 1 second. Is this not the, oh, this is
Is it-
PowerPoint. Sorry. I'm,
So if we just go to here, is that the?
sorry guys. Okay.
Maybe
I'll
just
go
click on
now.
this and then.
Oh, here we go. Okay, now you should just, right there.
¡Rescatando los anfibios!
Good.
Okay. Sorry about that. I tend to be a PC person. So my name is Allison Leidner, and I work with the NASA Earth Science Division, and specifically I work with our Biodiversity Research and Ecological Forecasting Applications programs, which are programs that directly fund researchers and decision support projects to help understand biodiversity here on planet Earth and also inform conservation activities. And so just as a note, at our headquarters activities is where a lot of our funded research happens. So I'm going to be really talking to you about NASA from a programmatic level. So NASA is an innovator for earth science and has an amazing set of observations, technology, and research that are at the cutting edge of science. But not enough people are using this for conservation. So what I want to talk to you about here is give you a sense of what NASA does for earth science and what's coming down the road for us in the coming years, and think about how we can apply that better to conservation. So this image of Earth at night powerfully conveys our human impact on the planet. And I know in the bright lights here, it's a little bit hard to see. But as you well know here at the Earth Optimism Conference, humans are the dominant species on the planet. And what the Earth Science Program and what the viewing Earth from space allows us to do is better understand how humans act as a global species and the impacts that this has on plants and animals on the land and in the ocean, and understand how we can better apply that to conservation. And so by taking this global view, by looking at it from the top down, and then working with our partners who look at everything from the bottom up, we can have a better understanding of how to integrate across scales and develop high-quality scientific data from a global perspective that can then be used for conservation. So this, there we go. NASA currently has 19, is there any way to dim the lights in this room? Can maybe someone try to take a shot at that while I'm talking? There's currently about 19 Earth observing satellites that circle the Earth. And you can see a variety of them here, and they take different kinds of measurements that are useful. So some of you may know that our satellites are able to look at temperature and precipitation. We can also make measurements of land cover. We can take a look at ocean salinity. We can take a look at productivity on the land and in the ocean. And it's this combination of observations that allow us to get a better understanding and piece all of this information together to understand how the Earth is interconnected, to understand land and ocean and atmosphere. And by looking at these interconnections, we can better use that to inform biodiversity. So what I want to give you a quick primer about first is how our satellites actually work. So our satellites don't just take pictures of the Earth. They collect sophisticated information about the light that hits the Earth and then is reflected back. And that's what our Landsat satellite does. So this Landsat satellite is a collaboration with the U.S. Geological Survey, as many of our satellites have domestic and international partners. And what the Landsat satellite does is circle the Earth once every 90 minutes, imaging a swath of that Earth. And then over 16 days, it visits every place on the Earth once, and we use that to develop an image of what is happening on Earth. And everything on Earth has a different spectral signature. And so when we are able to collect information in different wavelengths, so we can look at the visible spectrum, but we can also look at ultraviolet information, we can look at information that's in the thermal and in the microwave portions of the electromagnetic spectrum, we can actually get a sense of what is on Earth and how is it changing through time. And so it's this combination of information, and Landsat is actually a program that's gone back to 1972. So we have a 40-plus year record of what the surface of the Earth looks like. And this is an amazing record to use for conservation purposes. And the kind of information that we can get is what you see here. Oops, got a little ahead of myself there. What you can see here. And so this is the kind of high quality scientific data that can come from our Earth observing satellites. So what you're seeing here is vegetation on the land, productivity on the land and in the ocean, and you're seeing this through a seasonal cycle. And this data is combined from multiple satellites. And so what's really important here is that we can calibrate and validate our satellites against each other and combine all of this information, again, to provide this really sound understanding of how the Earth system is changing. But at NASA, it's not just about the observations we make from our satellites, it's also about the models that we use. And so we've developed a model called the ECHO model. And this is one of my favorite visualizations. I just think it's stunning. And this model takes satellite data and combines it with data that's collected from the ocean to look at how oceans circulate. And the colors that you're seeing are sea surface temperature. And this model is really great for me because it highlights how interconnected our Earth system is. You see how heat is transported from the Gulf of Mexico to Northern Europe to make Northern Europe warmer than it otherwise would be. Off of the tip of South Africa, you see those gyres that transport phytoplankton and zooplankton and also transport nutrients from the Indian Ocean to the Atlantic Ocean. And so it's this interconnected perspective that helps us understand what's happening at a local place, because many of you do conservation at a specific place or you work on a specific species. And so understanding the global context for that local place, but then also as we're able to develop higher spatial resolution information, as well as developing information that's collected repeatedly over time, we can also connect that to local places and scale back up. So you can't do conservation without this global perspective, and we can't look at this global perspective without tying that to the kind of information that people collect at local and regional, even continental scales, with information on the ground and in the ocean. So let me give you a couple of examples of what we fund people to do with this data. So in both of these cases actually take advantage of both research and our conservation applications projects. So what you're seeing here is a global map of forest cover that was developed using the Landsat satellite by Matt Hansen and his colleagues at the University of Maryland. And this global forest cover data set is a key component of Global Forest Watch, which is part of the World Resources Institute's new portal to provide information to decision makers in natural resource management. And with this global map, researchers at the Jane Goodall Institute looked at this and said, we can do even more than just looking at using information from where is forest and how is it changing through time. So the green areas are where a forest, and the blue areas are where we've seen forest gains between 2000 and 2012, and the red areas are where we've seen forest loss. So you can see the arc of deforestation in the Amazon and in other places as well. And the Jane Goodall Institute said, we have lots of observations about chimpanzees, and we know a lot about chimpanzee behavior. We know a lot about working with local communities in Tanzania and in the surrounding area, including Gombe National Park. So they all got together and collaborated to do this project where, with a grant from NASA to look at citizen science data. They armed local citizens with tablets to record where they've seen chimpanzees, when they've seen them. And for example, you can see in this bottom picture on the left is an example of a chimpanzee nest, to develop a more detailed understanding of chimpanzee habitat suitability in Tanzania, and then take that to local villagers and work with them to talk about identifying conservation areas. And it turns out that these areas are not just important for chimpanzee conservation, but because of the long history that the Jane Goodall Institute has in working with these places, they know that it's also important that reforestation efforts can help prevent landslides that affect local villagers in a variety of ways in addition to chimpanzee habitats. So Back to the Ocean is another project led by Helen Bailey and her colleagues at the University of Maryland Eastern Shore. And what they've done is they're concerned about ship strikes against whales, which is a significant cause of death among whales. And what the idea here is that there's a lot of shipping and naval activities off of the west coast of the United States that can hit whales. So if we have a better understanding of where we anticipate whales to be and when, we can avoid them. So they used ship-based surveys of whales combined with satellite data looking at sea surface temperature and sea surface height and chlorophyll to build predictive models. And so this is a screenshot on the bottom from their website just the other day about information from March and April as to where they expected the probability of certain whales to be, and then also how many they expected to be there. And by working with NOAA and other agencies, they can feed this information to shipping companies, to anyone who's interested in avoiding whales, and push that information out to help do conservation activities. So now that I've given you a taste of what we're expecting, sort of what's next for the agency, and this is a slide that will overwhelm you and it overwhelms you in ways that you may not even realize. So this list in this kind of lightish color, those are all of the satellites put up by NASA, again, many with international and interagency partners that are looking at Earth for Earth science. I think that's amazing. All of the yellow ones are the ones that we have coming up in the next decade. And that's even more phenomenal. And so what we're doing is looking at developing higher spatial resolution information, higher temporal resolution, and connecting that with previous satellites so that we can develop this long, scientifically consistent record. But we're also making new kinds of observations. That's part of the more of what's coming next. And so many of you who were at the opening plenary saw the talk from the International Space Station. And we use that International Space Station as an Earth observing platform. And one of the instruments that's going to be going up there is called JEDI. And that's in collaboration with the University of Maryland. And they're basically, as JEDI, you might guess there is a Star Wars connection, they are shooting lasers from space to look at vegetation. And so on the ISS, they can send out active information in the form of LiDAR, and then get a measurement, a 3D vertical structure of what forests look like. And so you can imagine how this revolutionizes our ability not just to look horizontally across the Earth's surface in 2 dimensions, but really add that 3rd dimension. So you can understand forest degradation. You can understand why birds and frogs and other species are using specific areas and why, and get this better understanding. Another instrument that's going to be going up on the International Space Station is an instrument called EcoStress that's looking at the effects of drought on natural ecosystems as well as agricultural systems, and getting a better sense of how water stress affects vegetation productivity, which is going to be particularly important in the light of climate change. We have other satellites. We have NISAR, which is an active radar mission that's in collaboration with the Indian Space Research Organization, ISRO. And so this is just giving us an amazing wealth of observations. But as part of this overwhelming amount of data, I like to say that scientists have a love affair with data. We always want more of it, but we get a little overwhelmed because there's a lot of it and we don't know how to process it. And so one of the uninteresting looking things, but very important things we're doing is developing an onboard processors that help process the data on satellites to help get it back to Earth, because we're collecting more and more and more data. But if we don't get it back down to Earth and get it out to people quickly in a format that's usable, it's not as great as it could be. So NASA has about 19 terabytes of Earth observing data in its records right now, and we add, I think, something like 12 terabytes a day. Just for reference, 19 terabytes. The Library of Congress, if you digitized it, would be about 10 terabytes of data. So that's an amazing amount of information. And so what we're also looking to do is help develop more analysis-ready data and what we call higher-level products. And so this is a way to get information to you, the biodiversity research and conservation community, in formats where you don't have to be an expert in remote sensing science in order to use that information for your own purposes. And so by working with our international partners, we can identify ways to develop these products that are really useful for you and where you can take your conservation expertise. And just as another plug for NASA, all of our data are free and open access. So anyone at any time can come in and download any of our data to do any kind of research or applications that they want. And so we're really interested in hearing from this community about what is it you want. And that's part of my job at NASA is I work with conservation NGOs. I work with our international partners to get a better understanding of what are your science needs so we can help tailor our research efforts for that. So we have a lot of exciting stuff coming up. We've done a lot of great work, and I'm happy to answer any questions later on, or we have somewhere in the back, someone found them finally, some NASA brochures about remote sensing. And so hopefully we'll pull them out if you want to get some more information about remote sensing itself, or if you want to hear some more conservation stories that NASA has helped support. Thank you.
See if this, awesome, it works. So I'd like to invite our panel up here and we'll have a little discussion. And I've actually boiled down what I want to ask of you guys to one question so that we can quickly open it up to Q&A, because I think we have about 15 minutes left in this room. So panel, my one question is, what is the one aspect of the work that you're doing that sort of is emblematic of the larger narrative? Like, what's the one thing you would tell people to get them really excited about the work you're doing?
I guess emblematic of the many problems that our planet is facing and the threats facing many ecosystems and many species, I think the one kernel of optimism I would provide is that with science and technology, new solutions are being developed. And if we can continue to support science and not deny it, that there's going to be a lot of advances that hopefully will continue to make this planet a better place. So I guess I'm an optimist at heart, and I'm pretty happy to be here at this meeting surrounded by fellow optimists.
So for me, we can see the problem. It's kind of like the guy picking up the trash on the beach. We can see the problem, and we can do something about it. And there's not going to be much point in trying to find a cure for a disease unless you have the actual animals to use the cure on. So it's a no-brainer for me, and it's, we're operating at a scale of 12 species on a single project. I think it's pretty awesome.
Following up on what David said, I think that some of the solutions to climate change are going to come from scientists. They're going to come from engineers. We are not giving up our lust for fossil fuels, and we're not changing anything. So we're going to have some technological fix that's going to help accelerate the removal of CO2 from our atmosphere. But I think a lot of the techniques that we're working on can actually help save coral reefs. And so it's establishing marine parks, but it's also using this technology to enhance those marine parks. So I think we actually have the ability to save a major ecosystem in our ocean, and to be very optimistic about that future.
One of the exciting things from our perspective is that we don't always know how our observational data and model data are going to be used. And so by making it publicly available, you all are free to go in and find solutions we would never think of, because you have so much greater knowledge of the specific locations or the cultures or the species or the threats to the environment that are occurring in a place, and apply that information. So I think it's just that creating an ecosystem for, innovation, for thinking outside of a box in a way that, remote sensing scientists at NASA would never think of to apply it to conservation problems and solutions.
Awesome. Okay, so I'm picking up on a theme of optimism being directly tied to this sense of agency that we all share, I think. So does anyone have a question? I'd love to open it up. Yeah? Okay, if you could come up to the mic, anybody who has a question, just go ahead and start forming a line. That'd be wonderful.
Hi, this is about the chytrid fungus. Do you have a clue as to where that may have originated? And also, when you talk about a cure for it, does it affect all frog species physiologically the same, so that when you find one cure, it's a cure-all? Or do you have to tailor that to each specific species of frog?
So the first part of the question was, where did chytrid come from? And the truth is, we don't really have a very good answer to that question. We do know that there is a very pathogenic strain that has spread around the world called the global pandemic lineage, that seems to have originated in the Old World and has been spreading through New World frogs, much like smallpox affected Native Americans when the first white guys arrived here. So it does seem to be a novel pathogen that's spreading through naive populations of frogs. But every time we understand a little bit more about chytrid, we find, there's been chytrid in Brazil since the 1800s, but it's not a very virulent strain. There's been very early records of chytrid fungus from Illinois. So, but we're not sure if it's the global pandemic lineage that's associated with the decline. So we're getting more answers and more resolution to that. But to answer your question about will one sort of cure approach affect all frogs? Well, I think it's going to depend very much on the kind of cure. If it's a probiotics thing, it tends to be very species specific. If it's something like a vaccine approach, well, that's also different. If you're going to go for a climatic refuge type approach, where frogs are introduced into areas that are too warm for the chytrid fungus, it'll depend very much on the differences in behavior between the different frogs and how they regulate their own thermal temperature from time to time. So I think it's all very context dependent on the cure side.
So I just had a question about how long we can cryopreserve the gametes. The reason for asking is, do we have enough time? I mean, let's say the sea temperatures keep going bad and more species keep dying. Can we reintroduce them after we use some technological solution, like you suggested, to get the temperatures back down and the CO2 also back down? Do we have that much time?
I think there's two parts to this question. Theoretically, we can keep cells or germplasm in liquid nitrogen as long as it's maintained well for hundreds to thousands of years. So, but we haven't run that experiment. We've only been able to cryopreserve since 1960. But the material that has been taken out of the banks, like bull sperm and things like that, have the same viability as when they were put in. So I think it depends on how well it's maintained. And the second part is the changes we're seeing in places like the Great Barrier Reef, where reproduction now, when you have a bleaching event, reproduction is not just impacted for the next years, and it's often ripples through for many years. And so if we get another bleaching event, it's going to be stymied again. And so as the bleaching events get closer and closer, reproduction will be impacted more and more. And so it concerns me that we are impacting reproduction so much, and that is one of the major ways that we are going to get resiliency is through sexual reproduction. So I don't know the answer to that. I don't know how, we can only hope that we'll have another 10-year period of good weather, where we can get out and do some of this. And that's really the reason that we're moving in the next 3 years to doing the symbiodinium, which is not that temperature sensitive. It is a bit, and also doing fragments, because we can move through populations really quickly, and it won't be related to reproduction. I can do it throughout the year, and we can train people really quickly, and we can go around the globe and really secure that material. I'm hoping that material will go back out into the water rather robustly.
I think we have time for maybe 2 more questions, so if anyone is shy, you might want to make your way up. Cool.
Are there satellites that can penetrate into shallow water marine habitats?
Yeah, so we do have satellites that focus on oceanic conditions. So we can only do it right now in the photic zone, so areas where the light does penetrate. But we are able to look at ocean chlorophyll, which is important. And there are people who are looking at coral reefs, and they do that both from some of our satellite data, but we also prototype a lot of our instruments on airborne. So on our airplanes. And so by doing that, we actually do have some instruments. I forgot the name of them, but they have been imaging coral reefs. So there's a campaign going on in Hawaii right now called Coral that is using our Avarice instrument, which is a hyperspectral instrument. So it makes hundreds, if not thousands, of measurements in the visible and near infrared range, and they're able to map corals with that. So I don't know more of the specific details of the science behind that, but we do have satellites and airborne instruments that are able to look at coral reefs.
Also, there's people are using drones now to do some amazing images of coral reefs, and Stanford actually has some new filter. Yeah, they have some new filters that can strip out the water surface, and so you're able to get 3D images and really nice high-resolution images of corals using these drones with these filters.
Hi, thank you for speaking here today. My question is about what are some of the effects on the natural ecosystem you've seen from the disappearance of the amphibians that you described was so drastic, and is that part of your research and part of your end goal?
I see there's some other frog experts in the room who might disagree with me, but so a lot of these really locally endemic frogs that are very rare, when they go, they're just gone. The birds still sing, the water still flows, algae still grows, life goes on. That's just the forest is missing a key member of the community. I've seen a lot of people really stretch to say, okay, well, if the frogs go, frogs eat mosquitoes, mosquitoes spread disease, that's bad. If the frogs go, the snakes that eat the frogs disappear. And to some extent, those effects are true, and they're there. There are ripple effects. But at the end of the day, the rainforests have lost their voice. They've gone silent, and they're missing a key ingredient. And I don't think that trying to sort of characterize it in terms of ecosystem losses really does it justice.
I think we have 1 more minute if anyone wants to jump up, or I think we'll all be sort of sticking around afterwards. So if anyone has any questions, thank you so much. And let's thank the panelists, right?